Hi, today we want to talk about protein synthesis. Proteins are one of the most important elements made in cells and are made of amino acids. The production of these molecules is done in two steps: transcription and translation. In a cell, there are multiple elements involved in protein production, such as DNA, mRNA, RNA polymerase, ribosomes, and tRNA. Transcription is the first step, and it happens in the nucleus. In this step, a message from specific protein coding parts in the DNA is made. RNA polymerase is the enzyme that produces this message, which is called messenger RNA, or mRNA. This enzyme moves to the protein coding section of DNA and opens the strands of DNA. Then it would build the mRNA using complementary nucleotides. When finished, the single-stranded mRNA is released, and DNA returns back to its original form. Now that transcription is finished, mRNA needs to get translated so that protein is made. For translation, other elements are required like ribosomes and tRNAs, but they are in the cytoplasm and not in the nucleus. So mRNA will migrate from nucleus to the cytoplasm and prepare for translation. Both mRNA and tRNA are made of nucleotides, which act like codes and are complementary. mRNA codes are called codon, and tRNA codes are called anticodon. The matching of codon and anticodon is essential for translation and protein production. Translation is started when ribosome detects the start codon of the mRNA, which is usually AUG. Then the two subunits of ribosome get separated. mRNA enters the ribosome and gets locked between the two subunits. tRNA, or transfer RNA, is a type of RNA which carries an amino acid. Amino acids are the building blocks of protein, and based on the anticodon of the tRNA, the amino acid it carries is specific. As the start codon is AUG, only the complementary anticodon containing tRNA can enter ribosome, which is UAC. This codon codes for methionine, the first amino acid of the chain. Then ribosome moves along the mRNA and to the next triplet codon and waiting for the next matching tRNA to enter. As the next tRNA enters, methionine separates from the first tRNA and forms a peptide bond with the next amino acid. As methionine separates from tRNA, it sticks to the amino acid chain, and the empty tRNA leaves the ribosome. This process is repeated, and every time an empty tRNA leaves, the ribosome moves forward to the next triplet. Translation goes on until a termination codon is reached, which is UAA here. When ribosome moves 2 to this codon, a release factor enters and signals the termination of translation. Then the amino acid chain is released, also the mRNA strand and the last tRNA both leave the ribosome. Finally, ribosome subunits return to their initial form. The protein is now made and ready for folding to become functional, which we will explain in the next videos. This is the mRNA codon chart. Each triplet codon translates to an amino acid, unless it is a stop codon. There are 3 stop codons: UAA, UAG, and UGA. AUG is the only start codon, coding for methionine. There could be more than one type of triplet codon for a single amino acid, but there couldn't be more than one amino acid for a single codon. This is the end of protein synthesis. Thank you for watching. If you enjoyed this video, please don't forget to subscribe.